So the chromosomal basis of inheritance here. So Mendel gave the gene specific loci along the chromosomes and helped to look at that. And it's the chromosomes that undergo segregation and independent assortment is what we've got in here. So this is showing here that you've got Mendel's idea here with these particular genes along the chromosome. And then it's showing the chromosomes undergoing assortment in this picture, and here going through meiosis. So that what you can end up with is your gametes in here that may or may not have that gene, and how we get to that point. So when we're making those Punnett squares, what we do along the side is make the gametes and the possible combinations in there. So the law of segregation says there's 2 alleles for each gene that separate during gamete formation in there. And the law of independent assortment says that the alleles of genes on non-homologous chromosomes assort independently during gamete formation. So if they're on a homologous chromosome in there, that's not what he's looking at. He's looking at the non-homologous ones in there. So another researcher who did some studying on this was Thomas Morgan, and he was an experimental embryologist in the early 20th century in there, and he was skeptical of Mendel's ideas in there. So he studied the fruit flies, the Drosophila, and you'll notice a lot of labs in the book. There's a few different labs that talk about using fruit flies, and for doing genetic studies in a classroom, aside from the fact that they're fruit flies that you've got to keep alive for a few days and then you got to kill them and then you've got flies all over the place in here, they are good for that because they're prolific breeders in here. It doesn't take long to get a new generation of flies. Every 2 weeks a new generation of flies. They can double and you can have total infestation within a short time in there. They only have 4 pairs of chromosomes, 3 are autosomes, 1's a sex chromosome. So they're pretty simple. They basically breed. So when we look at the phenotypes in there, we have the wild type, which is the character that's most commonly observed in natural populations, and then the mutant type, which is the trait that's an alternative to the wild type. And it's just assumed that there was a mutation to create that mutant type in there. So Morgan studied the eye color on his fruit flies and observed a correlation between a particular trait and the individual's sex. And so this is showing his studies in there that you had offspring, the correlation in there with having certain sex having more likely to have a certain color of eyes in there. And this was how we really learned about sex-linked genes, which was that third little square that I put up in there. So when you do a Punnett square with a sex-linked gene, you're going to have the superscript just like you did for the codominance or incomplete dominance in there. And you already know how to do those Punnett squares. It's the same thing in there. But in humans and animals, the sex-linked is going to be important because in females you get 2 X's, in males you get an X and a Y. And you'll inherit one from each parent. So for a male, if something is on an X chromosome, he doesn't get 2 X chromosomes in there. So he doesn't have the second allele to have a possibility on there. If there is a recessive trait on the X chromosome, he's going to express that trait because it's the only one of those chromosomes he gets. Where with a female, if she's got a recessive trait on one of the X chromosomes, it won't be manifested unless the recessive trait is on the other X chromosome too. So those sex-linked recessive traits occur more in males in there. Short segments at either end of the Y chromosome are the only regions that are homologous with corresponding regions of the X chromosome. So most of it is not homologous in there. For humans, signs of the sex of the baby start to appear at about 2 months. Prior to that, the gonad development is generic. And that's because in 1990, well, this gene was there before 1990, but that's when they identified this one gene called the SRY gene that is your sex-determining region of the Y chromosome that is going to code for proteins that regulate other genes that cause male development. So what's going to cause you to develop as a male is having an SRY gene, regardless of which chromosome it ends up landing on. Generally, the SRY gene is on the Y chromosome. But in about 1 in 10,000 cases, you can have crossing over and the SRY gene end up on an X chromosome. So if you have an SRY gene on an X chromosome, even though chromosomally you would be a female, you would develop as a male. And so we have about 1 in 10,000 people out there of each sex that are chromosomally the opposite sex from what they developed as. And they don't usually know it until they find out they're infertile in there. So the Y chromosome is much smaller. It has 78 genes on it that code for 25 proteins. Some of them are duplicates, and only about half of those are expressed in the testes in there. So it's a very small chromosome. It's also one that is about 1,000 times more likely to have mutations in it than an X chromosome is. So a lot more variations in that Y chromosome. You could be if your SRY gene ends up on the wrong. And you would never know until you go to make babies and that doesn't work. So the sex-linked genes is going to be a gene link that is located on either of the sex chromosomes in there. Y-linked genes are going to be on the Y chromosome, and they're passed from fathers to sons. There's very few genes and very few Y-linked disorders because there's only 78 genes on there. But if it's a Y-linked disorder, you cannot get it as a female. It only occurs in males, and he inherits it from his dad. So for the X-linked genes, there is about 1,000 genes on the X chromosome. And they'll have a different pattern of inheritance in males and females because females inherit 2 X chromosomes, males only inherit 1 on there. So the X chromosome, a lot of the genes are completely unrelated to sex. Fathers will pass X-linked alleles to all of their daughters and none of their sons, because if they pass an X, you turn into a daughter. If they don't pass you an X, you turn into a male. And there, the mother can pass X-linked alleles to both sons and daughters. X-linked recessive traits are expressed in females only if they're homozygous. So one we know of is like male pattern baldness, and we call it male pattern because it occurs more likely in males. That's on the X chromosome. So if he inherits that from his mother, then he's going to express that. Females can also inherit it, but they have to inherit it from both of their parents. And so where it's males are absolutely going to inherit it, it's less common in a female because she has to inherit it from both. Yeah.
So doesn't the male determine what the sex of the offspring is?
Mm-hmm.
Why is that?
Because the male is the only one that has the Y chromosome. So when his gametes form, you're going to have one X goes into one sperm, one Y goes into the other sperm. So if you get the X sperm, you turn into a female. If you get the Y sperm, you turn into a male. So if they don't like the sex of the baby, it wasn't your fault what you got in there. So the male that has only one locus, any male receiving a recessive allele from his mother will express the trait in there. So Duchenne's muscular dystrophy is X-linked, 1 in 3,500 males in the U.S. This is going to involve progressive weakening of the muscles and loss of coordination. It's rare that they will live past their 20s. They're missing a protein called dystrophin. And so with this, because of the males having it, a lot of times because they don't live that long, it doesn't get passed on from them, but it does get carried by the mothers in there. So hemophilia is an X-linked recessive gene where you're missing one or more of the proteins for clotting blood. So again, going to show up more in males than females. So females are going to inherit 2 of these X chromosomes, but you don't need to have both of them be active. The humans are adjusted to having one of them be active. So one of them is going to get inactivated during embryonic development. And the inactive one is going to form what we call a bar body in there, where it just condenses in there. And then when you go to produce eggs, the bar body regenerates itself in there and gives you the second X chromosome to pass on to your eggs. So as a female, all you have are X chromosomes, and that's what you're going to put in all of your eggs in there. So you can kind of end up with a mosaic of the 2 cell types in there if what you actually inherit are a trait, 2 different traits on an X chromosome in there. For each individual cell, it's going to be, each cell is going to determine which of those X chromosomes becomes the bar body. So if it's the chromosome dad in one cell, it may be the chromosome you got from mom in the other cell in there. So you can actually have a mix of the 2 characters in there from the X chromosome because of the bar bodies in there. It won't be that you necessarily inactivate all of your X chromosomes from dad. Once you inactivate it from that cell, though, all of the cells that are derived from that one with it inactive will be the same. But initially, when that inactivation happens, it's independent on there. So the X inactive specific transcript is going to be the only active chromosome that will become the bar body in there. So you modify the DNA and the histone proteins in there to create that bar body. So we have other genes that are linked together, and what makes them linked is being near the same location on the same chromosome. So when you have crossing over in there, some of the genes are more likely to all cross over together. When you have that happen in a linked gene in there, what we look at is, because we know that things are going to assort independently in there, it's a linked gene if it occurs more frequently with another trait, more than 50% in there, because they are crossing over together. So the traits are inherited in specific parental combinations because the genes for the characters are on the same chromosome in there. So when you have the genetic recombination, you produce offspring that have combinations of traits that are different from each parent in here. So when you have the genetic recombination of unlinked genes, they're going to assort independently. And the parental types are going to be the phenotype of the parental generation. The recombinants are the new combinations of phenotypes that are in your offspring. So 50% frequency of recombination is what you're going to see for any 2 genes located on different chromosomes. So you've got a 50% chance of them occurring together. So they're not linked. If they occur together more than 50% of the time, then they're considered linked in there. So crossing over gives you the recombination in the linked genes in there. And while it's replicated, the homologous chromosomes are paired during prophase 1 in there. And so you can have, during the recombination, it's more likely to take that other gene with it because it's close in location. So this is kind of showing it here, where if you've got a couple of genes that are in this region, when this section crosses over, you're going to have both of those genes will travel together. So we consider them linked genes. And the closer they are, the more likely they are to travel together. So you can use that information to make these genetic maps in here, where you have a list of the genetic loci along a particular chromosome. So the recombination frequency looks at the percentage of recombinant offspring, and they will infer the distance between the genes on the chromosomes based on how often the linkage occurs together in there. So the further apart two genes are, the higher the probability is that a crossover will occur between them and separate them. So the linkage map is based entirely on the recombination frequencies in there. So 1 unit is defined as equivalent to 1% recombination frequency in there. So when we map out things genetically like that on a chromosome, it's not necessarily giving an actual physical location in there. They will get mapped out based on how often we look at how often they will cross over together in there. And that's part of how we've mapped them out in there. So cytogenic maps are going to locate genes with respect to chromosomal features in there. When we look at all of these different types of maps that we can make together, we know that the order of the genes on the chromosome is the same, but depending on how you create the map, the space between the genes is different in there. So that's what is not necessarily going to be as accurate as in determining is the space between the genes. So another thing that can happen, altering the chromosome number in there and some genetic disorders. If you do large-scale chromosomal alterations in humans and animals, especially the mammals, usually it's going to spontaneously abort. We don't do well with extra chromosomes, and neither do the other animals out there. Those that survive are usually going to have developmental disorders. The most common one is trisomy 21 Down syndrome in there, where they have an extra chromosome. So what happens in this is you have this non-disjunction in here. Members of a pair of homologous chromosomes don't move apart properly during meiosis 1, or the sister chromatids fail to separate during meiosis 2. So it can occur in either place where these don't separate or they don't separate here. So what you end up with is aneuploidy, which is just an abnormal number of chromosomes. Monosomy, or a monosomic individual, has a fertilization that involves a gamete that has no copy of a particular chromosome in there, so it's going to have a missing chromosome. Trisomy has an extra chromosome. One of them is in triplicate. Polyploidy is an organism with more than 2 complete chromosome sets in all the somatic cells. Polyploidy is common in plants, not in animals. So those are the things that can occur where you get extra or missing chromosomes in there, those disorders. When you have the chromosome itself get altered in there, you can delete fragments of the chromosome in there, and that will cause what we call a deletion mutation in there. And sometimes when that piece gets deleted, it doesn't just disappear into nowhere. It ends up attaching to the end as an extra segment on a sister chromatid in there. So you end up with one that's too long and one that's too short in there. It's the duplication on the sister chromatid then. You can have inversions where a fragment of the chromosome comes out, flips over, and goes in backwards in there. One thing we know that causes inversion mutations is asbestos, that it will have chunks of chromosomes get cut and go back in. Translocation, where a fragment joins to a non-homologous chromosome in there. So in males, large deletions in the X chromosome will tend to be lethal, where they will tend to survive if they have a duplication or a translocation. They'll survive, but they will be harmful in there. Just want to see where we are time-wise. Okay. So some chromosomal alterations, Down syndrome or trisomy 21. This is 1 in 700 children born in the U.S. have an extra 21st chromosome, tend to have characteristic facial features. Short in stature, they have a lot of correctable heart defects, developmental delays. They have an increased chance of leukemia. Their chance of leukemia is actually really high in there. Alzheimer's disease, it will tend to have an earlier onset in them. A lot of times they'll start to get Alzheimer's in their 20s or 30s. Decreased risk of high blood pressure and atherosclerosis, increased frequency of, you see it with increased frequency as the mother gets older in there. So older moms are going to be more at risk for having this. And it's one of those curves that, it starts gradual, but when you get into your 30s, your risk goes like this, and they're for having a baby with Down syndrome in there. Most of them are still born to younger mothers because they're the ones that have most of the babies, but your chance of your baby having Down syndrome is much greater as you get older. The aneuploidy of sex chromosomes, these appear to have less effect in the sex chromosomes than in the autosomes in there, being that the Y chromosome has relatively few genes and the extra X chromosomes get inactivated as bar bodies. They figure that's why you're able to survive with those a little bit more. So Klinefelters, this has an extra X chromosome in here. It's 1 in 500 to 1,000 live male births. They have male sex organs, but the testes are small, they're sterile. The extra X's are inactivated, but sometimes you'll see some breast enlargement and female body characteristics in there. Most have a subnormal intelligence in there. More extra chromosomes you have, usually the lower the intelligence is. The XYY, the super male, is 1 in 1,000 males in there. They have normal sexual development, no well-defined syndrome, but tend to be taller than average. There have been some studies out there that suggest with higher testosterone levels, they're more prone to violence. There are studies that argue that. One of them, I looked at it, and its argument was, well, they only beat their spouses. And I'm like, okay, so that's not violence in there. I just kind of looked at that and I was like, well, that's kind of interesting in there. And then I never looked any further on it. But there have been some ideas of that out there. How true it is, I guess probably depends on what you consider violence. So the XXX in here, this is 1 in 1,000 females. These are usually healthy with no other unusual features other than taller than average. More risk for learning disabilities, but they are fertile in there. So Turner syndrome is the monosomy X. This is the only known viable monosomy in humans in there. And phenotypically female, but is going to be sterile. The sex organs do not mature. Girls with Turner syndrome can develop secondary sex characteristics if they're given estrogen therapy in there. Normal intelligence levels. Part of it depends on the estrogen therapy needs to happen at a certain time frame in there to really kind of help give them a normal life, and whether or not the parents will do that. Sometimes parents will be kind of like, I don't want my little girl to grow up. And if you've got an 18-year-old that has never gone through puberty and stuff like that, you're probably not really doing her any favors in having a normal life in not getting her any hormone therapy, but it does happen in there. Some other ones, some structurally altered chromosomes. Cri-du-chat, this is a specific deletion in chromosome 5. They're severely intellectually disabled in there. They'll usually have a small head, unusual facial features, and they make a cry that sounds like a distressed cat, and they usually die in infancy. So the CML in here is your chronic myelogenous leukemia in here. This is a chromosomal translocation, and it occurs with a large portion of chromosome 22 in there, with a tip from fragment a fragment tip of chromosome 9 in there, and it produces what they call the Philadelphia chromosome in there. And so it's uncontrolled cell cycle progression. And with leukemia, that's where it will show up in there, because that's where you're going to have a short cell cycle, is you're constantly replacing blood cells in there. And if you start changing the cell cycle in there, it's more likely to show up in there because it goes through the cell cycle more frequently than other places in your body. So there are some patterns that are exceptions to standard Mendelian genetics. One of them is genomic imprinting. And this is a variation in the phenotype depending on whether or not the allele is inherited from the male or female parent. A lot of times it's just adding on a methyl group to a cytosine on one of the alleles is all that's happening on a biochemical level. But it occurs during gamete formation and can result in silencing a particular allele of certain genes. And so they're imprinted differently in the sperm and egg, and the zygote expresses only one that's inherited from the male or female parent. Kind of an interesting thing with this is they've been looking at it, and they think this may have something to do with homosexuality in there, that you end up with a gene that is imprinted from the opposite parent in a different way than what happens normally in there, that may be part of it. And so it'll be a while before we know anything more on that, but that's one of the places where we look and think that might be an implication of genomic imprinting in there. So organelle genes, you do have a few genes that are outside of your nucleus and not part of the chromosome, some cytoplasmic genes in there. Most of them are going to be in the mitochondria. In the plants, they're in the chloroplasts and the other plastids, but they all contain a small circular DNA. These do not display Mendelian genetics because when you inherit the organelles, you have the male and female cell come together for fertilization, and what's going to happen is the sperm is going to drop off the chromosomes in there, the DNA, but it doesn't give any of the organelles to that cell. All of your organelles start out from your mother, from the egg cell. So all of your mitochondrial DNA actually comes from your mother in there. So that follows a different pattern in there. You are actually slightly more like your mother than your father for that reason in terms of what you got for genetic information in there. So what these mitochondrial genes tend to make up are protein complexes of the electron transport chain, ATP synthase, things that are going to affect metabolism in there. So I looked at this and I went, this is not good, because from my mom's side of the family, not athletic at all, and everybody is fat. Every last one of them is fat. And that's where I got my metabolism DNA. So that just means I get to work harder at not looking like them for the rest of my life in there. And every time I am out doing a marathon at the end and it's hurting and everything, I'm like, that damn mitochondrial DNA. I know who to be mad at now. So there are some diseases that we look at that you're more susceptible to due to mitochondrial DNA: myocardial myopathy, weakness, intolerance of exercise, muscle deterioration. So yeah, at the end when it hurts, I'm blaming her. And then what they call Leber's hereditary optic neuropathy in there, sudden blindness in a person as young as in their 30s or 40s. There's 4 mutations so far that are found to cause this disorder in there. So yeah, they think that it's linked to, at least linked in some way to, diabetes, heart disease, and some other disease like Alzheimer's in there with the mitochondrial DNA. So that is 15.